When we look at the process of metabolism, we can talk about two separate states, an absorptive state or a post-absorptive state. So in the absorptive state, you could also think of that as like a fed state. But remember, this is a continuum. So an absorptive state occurred typically after a fasting state, right? So this fed state occurs after a fasting state had occurred. So in our absorptive state, carbohydrates that are consumed are broken down into simple sugars, and that happens via hydrolase enzymes on the brush border of small intestines. And this is just kind of giving you context of where we are. This is organs that are outside of the intestines. This is not the enterocyte. This is liver, brain, muscle, fat tissue, and our red blood cells. So just to give you some context here. Once the simple sugars are moved into our enterocytes, they do so using a sodium symporter. So again, sodium is moving down its concentration gradient and it's taking something like glucose with it, down its concentration gradient from the lumen of the gut to the actual enterocyte, the cell. Proteins get broken down into amino acids, and they're also moved into enterocytes using a similar sodium symporter. And both amino acids and glucose will enter into the bloodstream, and they'll travel then to the liver via the portal vein. And so we can see here glucose being able to travel from the blood, which is just kind of the space, same with amino acids traveling from the blood into the liver, and this is via portal circulation. From here, in this absorptive state, we'll see increased glucose and amino acids are present after a meal because we just took food in. And that's going to lead to insulin to be released from our pancreatic islet B cells or our beta cells. And when that occurs, we're going to see a couple actions take place. Insulin is the main hormone that's directing organs and tissues and cells to use nutrients during this state. And all tissues are, regardless of insulin, are going to increase their uptake and use of glucose and amino acids under the influence of insulin. But there's a couple tissues that don't require insulin to take up glucose and different kind of molecules. The liver's one of them. The liver doesn't require it because things can travel via portal circulation to the liver from the digestive tract, although the liver can be influenced by insulin, but doesn't require it. Red blood cells do not require insulin. Red blood cells are going to only use glucose as fuel to create ATP. And then they get stuck with pyruvate, which they break down to lactate as a byproduct. And then the brain prefers to use glucose as fuel and will continue to use glucose so long as there's enough glucose around. Again, going to, the typical process of forming ATP because there's all the machinery needed in neurons to do so. The liver, during this absorptive state, is going to monitor and regulate how much glucose and how much amino acid levels are in the blood via how much is coming in the portal hepatic vein. The hepatocytes, our liver cells, are going to convert glucose to glycogen in this stage using glycogenesis until the liver stores are met. So we have so much glucose, we can create it in our storage forms. We also are going to take glucose down its process through glycolysis to the Krebs cycle to oxidative phosphorylation to create energy. So kind of your classic aerobic respiration here. And hepatocytes can also take acetyl-CoA and they can break them down and form fatty acids that get essentially transported in as triglycerides, glycerol plus fat. And then they travel via our very low density lipoproteins or VLDLs to be taken up by our adipose tissue, where it then unpackages that VLDL into fatty acids and it's stored until it needs to be utilized for fuel. And this is essentially lipogenesis. Our hepatocytes also are using amino acids for protein synthesis, and then they can also export excess of them within the circulation if they don't need it. So this is kind of our absorptive state, or some would say maybe our fed state. We can see these amino acids being used to create proteins. We see glycogen being created. We see fat being created and stored. So we're creating energy, lots of ATP being generated, and we're creating storage forms of energy in all of our cells. Now, insulin is the main regulator of our absorptive state. It targets liver, muscle, and adipose tissue, and it's going to stimulate glycogen synthesis and triglyceride formation. Now, the brain and erythrocytes are insensitive to insulin and they do not respond to it. They will take up glucose regardless of insulin's presence or not. And liver will also do the same, but liver can be sensitive to insulin. So it'll take up more insulin if it is or take up more sugar, more glucose if it's influenced by insulin. But the brain and erythrocytes are not. They are essentially stable. Okay, so then moving into our post-absorptive state, at the end of the absorptive state, our gut cells, our enterocytes, stop taking in glucose into the hepatic portal circulation, but our peripheral tissues will continue to take up glucose. They'll continue to use glucose uptake. Glucose then levels in the blood start to decrease because we're not getting any more in through our small intestine, but we're still using it in the body. And that's going to start to stimulate glucagon and epinephrine levels, which are going to encourage, instead of bringing glucose into cells to create stuff, kicking glucose out of cells. So our hepatocytes start glycogenolysis, essentially breaking down glycogen into glucose. So we have a breakdown of our glycogen stores instead of creation. Liver stores then start to become more and more depleted, and gluconeogenesis is stimulated by glucocorticoids like cortisol that are released from the adrenal glands. So we start to go through this reverse process to again get more glucose. We'll see any circulating fatty acids that are present and glycerol are also used in this process of gluconeogenesis. So you can see fatty acids being brought in to create acetyl-CoA, which then can, I mean, be used for lots of different reasons, same as glycerol broken down. And we're seeing that we're starting to utilize our fat stores, creating fatty acids for energy and then for release to circulation. The longer we stay in this post-absorptive state, the more the liver starts importing amino acids from our muscles to essentially push them through gluconeogenesis, as well as starting to synthesize ketones or ketone bodies using ketogenesis. And then we see this increased level of circulating ketones and lipids, um, and this decreases the peripheral tissue's reliance on glucose. Now, an important note, neurons and red blood cells rely on glucose metabolism primarily, and red blood cells are actually only able to use glucose as their fuel source. So those are the two tissues that are going to continue to use glucose until the very end. And neurons, once there's not adequate glucose supply, neurons will allow our red blood cells to utilize whatever glucose we have, and neurons can survive off ketone bodies. Glucagon and epinephrine are the major regulators of the post-absorptive state. They are increased in the fasting state and they target liver, skeletal muscle, adipose tissue to break down glycogen. And we see a release of fatty acids and amino acids being stimulated by a decrease in levels of insulin and increases in levels of epinephrine. If fasting is prolonged, glucagon and epinephrine levels will increase, and we'll see lipolysis will form excessive acetyl-CoA, which is used for ketone synthesis. There'll be an increased level of lipids and ketones. Muscles will start to use fatty acids for fuel, and the brain will utilize ketones for fuel. Proteins are spared until the very end, as ketones can be used instead of glucose. And it's of note, erythrocytes will continue to use glucose as their major source of energy. We'll stop here. When we come back, we're going to kind of go into some of the biochemistry and some of the background behind hormones in these different glandular areas.